Good afternoon, everyone. My name is Dr. Lauren Juskovich, and believe it or not, my background and my PhD was specifically in water quality. So I am a huge advocate about water and, kind of playing off of what Nick said that, um, healthy fish are happy fish. I would like to say that clean water makes healthy fish, and therefore healthy fish are happy fish. And so let's talk about cleaning that water, what type of water quality, and breaking down that web as to those different pieces or so. And so I guess in order to do so, I really just want to bring it, and I know a few of you might have heard this talk from me a few times, and hopefully something else new resonates. But if you're going to start an aquaculture or fish farm, who are the first three people that you should hire? And really, or if you had a pet, say, what are the first three things you should be thinking about? Well, one, fish biologists know what they need to eat, where they should live, know that tilapia don't really survive here in Michigan because it gets too cold, things like that. And so, basic care of fish. And then two, vet when they get sick, right? We want somebody to be able to help treat them and make them better. And then I would say a third would be a water quality expert, somebody who knows that, okay, so there's something that we don't see but we know something is happening with our fish. What can we do that might not be so acute that we need a vet, but we want to improve our production and make sure that we have great survival techniques. And so, let's, as Myron was talking about in terms of, the pathogen is always there, then there's a circle of the host, and it really needs to be a favorable environment for those three to work together to create a disease. And so what we're specifically talking about in water quality is kind of flipping the switch of when does that pathogen become disease or not. And so those are what parameters we would like to control. And so, just like a broad overview of like what and when we should monitor. These are some of my recommendations based off of research. And daily, you kind of want to go around and check your pH, your temperature, your dissolved oxygen or DO, conductivity or salinity if that applies, and then also your nutrient inputs and outputs, such as your feed, your sludge, erosion, and the fish themselves. And then in weekly, kind of like more secondary in terms of influence would be your nitrogens and phosphorus. And that would be total nitrogen in the form of total ammonia nitrogen, nitrite, nitrate, and then total phosphorus or soluble reactive phosphorus. And we'll talk a little bit more throughout this talk about these specifically. And then more monthly and quarterly are your total suspended solids and your biochemical oxygen demand, or your BOD5. And so, we want to look at, okay, so how do we know we're actually being successful or responsible with our water quality? Here's another kind of overview of what parameters to maybe look for, specifically more for ponds or raceways or RAS, based on the best aquaculture practices, or BAP. It's a third-party certificate for being environmentally friendly. And so this is something that you can be certified with, and these are kind of their goals or good targets to kind of go with in terms of those parameters, so that you're keeping that water clean and fish healthy. And so something that is specific to our region, especially with flow through, is the NPDES. And this is a National Pollutant Discharge Elimination System permit. And so this was established after the Clean Water Act. It gave the EPA kind of guidelines to develop, okay, so how much can you be discharging into a waterways? We want to clean up our waters. How can we prevent this and monitor it? And this was a permit that affects certain farmers based on how much you produce, if you need this or not. And so it really, is associated with the discharge of pollutants from a point source. So that would be an aquaculture facility from, say, a flow-through system into natural waters, not necessarily a non-point source pollution, which would be like seepage from a pond. And it's really designed to protect the public health and the environment, right? And so we want to avoid water quality degradation because your fish won't be healthy in unclean water. So we want to keep it as clean as possible, as well as being responsible for our surrounding environment as well. And can be relatively strict guidelines from the EPA, excuse me, and then the state can actually use either those guidelines or be more strict by a state-to-state basis, and that's usually housed in a water quality or water environment agency. And so, for example, some of the more common concentration-based criteria for these discharge permits include pH, dissolved oxygen, biochemical oxygen demand, and total suspended solids. So let's talk a little bit more about, okay, you're going to take water quality samples. Really, you don't need any special tools, as you can see here in the picture, but really just take any kind of bottle, water bottle that we're done drinking out of, and rinse it 3 times with the water sample, and then fill it out without leaving any air at the top. We don't want air. If you're sending it to a lab, ship it on ice and in a cooler or insulated cooler, concealed from light. That light will also make it more reactive with the algae that's still in there. You could also do composite sampling, which is kind of taking samples from multiple areas in your water. For instance, you don't really want the bare minimum surface water, since it's really reactive to the air, or down too far, or over here, over there. And so mixing a couple samples together is a composite sampling. And it really depends on the type of system as to where might be best to take those samples from. But, okay, so you're like, Lauren, you're throwing out all these parameters, and really I've taught whole classes of this, but why right now should I worry about water quality? Well, one, if you're growing fish, that's your money, that's your livelihood. You want to keep the product alive. 2, your reputation, it's your brand. You want to put out a nice, healthy product, whether that end product is for food, bait. You want people to kind of come back and eat and feel safe with that product. And so in order to keep fish alive and your business going, you should care about water quality and learning a little bit more. And so when temperature and pH are just like, oh, we just monitor it with this probe, and it just, it's something that we do, but we don't really know too much about it or what it impacts. But really, temperature and pH, the biggest factors are really the protein conversion efficiency for fish. So this really affects your feed conversion ratio. And so how much of that feed are the fish using to create more fish rather than going into the environment, right? So how much of that feed is being converted, then determines which species can actually be produced, right? So we want to make sure that our environment is suitable for what can be grown. It also helps determine your growth rates. Is it going to take you 3 years to get to market size, or 1 year? And it determines the balance of these other water qualities. So kind of that web, right? The systems thinking of water quality. Water quality is not static. It is continuous. And so when you take samples, you are taking a snapshot in time of what those parameters are. And then over time, they change intricately, right? And so it's that balance of all of them. And so, dissolved oxygen, I definitely agree, is probably the #1 go-to. A lot more fish actually probably die from dissolved oxygen than most farmers realize. But it's not that feed plus fish equals bigger fish. That's not the equation that goes on, mostly because you're in a system that's constrained by oxygen. It's like putting a pig in a space rocket out into space and you're growing it, right? Oxygen becomes a limiting factor. So that's something that you have to contend with. It's not like an open air farm or your free range chickens and things like that. Well, the feed does not only feed the fish, but it's also feeding. So those nutrients are being supplied to the phytoplankton, which heavily determines the amount of dissolved oxygen in the water if you're not doing a full recirculating system. And so small organisms also use more oxygen per unit of weight than larger fish. And so they're a little bit more sensitive to these changes, because kind of thinking of like surface area ratio. Increasing temperature increases consumption rate of dissolved oxygen. And then feeding requires a lot more oxygen. So when you're feeding, and if you were actually measuring your dissolved oxygen while you're feeding fish, you'll realize that the oxygen's depleting a lot faster during that feeding event. And then fish swimming against the current will actually increase their oxygen use, and as the current velocity increases. So while they're kind of using that energy and that exertion, right, kind of think of us running, they're breathing out more CO2, so they're inhaling a lot more oxygen at the same time. I just kind of wanted to briefly show this, that, you know, without the presence of oxygen, that's when we start looking at fermentation zone. And so here, if we were looking at, say, like the side picture of a pond, we have here the water area where, you know, carbon is constantly being pushed into CO2 with oxygen. But once you hit a few centimeters below the top surface, or sorry, millimeters, a few millimeters below the top sediment water interface there, oxygen's not present anymore. And so where does fermentation happen, or respiration? Where does that oxygen come from that's not there anymore? And so that's where we start seeing different forms, such as denitrification, such as with nitrate, iron, and specifically more of a concern would be hydrogen sulfide and methane. And so that's something to consider if you're using groundwater in that interaction as well. And so, you know, talking about oxygen and those daily requirements, now what about those weekly ones? And one form is nitrogen. And this is more coming to be the more limiting factor in terms of increased stresses. And it's really just knowing how to manage nitrogen, and nitrogen just being this continual basis of different forms of nitrogen, and each form reacts differently to fish. And so the forms change based on how much oxygen you add or remove. And so nitrification is just oxidizing the nitrogen element, while denitrification is removing oxygen. And so if we wanted to go from elemental nitrogen in the water to nitrite, we could add an aerator. And then if we want to convert nitrite to nitrate, we would just keep that aerator going, and so on. So very simple terms there, but so that's the idea between nitrification and denitrification. It is important to note that they are not necessarily a 1-to-1 ratio, so they do not always overlap exactly chemically. And so if we look at nitrification a little bit more, oxidation where energy is released. It uses two specific types of bacteria, Nitrosomonas and Nitrobacter, and organisms take energy to make organic matter from carbon. And so it takes 2 units of oxygen. And so you could actually calculate, that's what I hope to show here, is you could actually calculate how much calcium carbonate is produced, or alkalinity, or taken away based on your amount of nitrogen that's present in the water, or how much oxygen is needed for you to kind of get past a certain element. And so talking about ammonia, you know, NH3 is toxic, or the unionized ammonia, but we can't really measure it specifically by itself. We need to measure it as total ammonium nitrogen. And that toxic proportion of that equation increases as temperature increases. And so every time a temperature increases, it doubles the proportion of toxic ammonia. But pH increases it by 7 times. So pH is a lot more of an influencer than temperature. And to kind of see that at an example, say you have water at 20 degrees Celsius and 30 degrees Celsius with a pH of 8, we can see that out of the proportion, the unionized ammonia is more at 30 degrees, right? Higher temperature. Now, what if we raised the pH? Well, we could see that greatly influenced the concentration, even more than just a 10 unit degrees in temperature. And so it really depends on these other parameters, that wonderful intricate web. But it's really difficult to prove if something died from ammonia. It really just causes stress, which makes the fish more vulnerable to get a secondary infection, which is when that pathogen becomes a disease, right? And so, yeah, there are tables out there. Happy to provide those as resources if you want to be able to calculate using your pH and temperature how much of the toxic ammonia is present. And there are some safe levels listed there. And so I'm gonna show a little snippet of my PhD work, and it's so clear how much aeration can impact nitrification. It's shown in this graph, and I just geek out about it. But so here on the x-axis are dates, and on the y-axis is the total ammonia nitrogen level. So total, right? Not just toxic, but as I studied basically additional aeration in a split pond system, and four ponds had extra aeration, 4 ponds were the control, so they had less aeration. And we could see in year 1, where there was not extra aeration, they were kind of going the same. And then by year 2, we added that additional aeration right here around springtime in May. And the total ammonia nitrogen, or TAN, stayed much lower in those ponds that had additional aeration in their system than those that did not have aeration. And that's because that total ammonia nitrogen was being converted more rapidly to nitrite and nitrates and going through the nitrification process. And so this is really cool to kind of see how much of a change just adding aeration can do to make your system healthier. So, okay, we got rid of the ammonia. Now it goes to nitrite. Well, nitrite is also very toxic to freshwater fish. It actually creates brown blood disease, and it turns to brown blood due to the creation of metahemoglobin. So it doesn't really carry oxygen anymore. And so you'll see these, it's called hamburger gill disease. And so they look like rotten hamburger gills. It really depends on the nature of the water. So this is more influential in freshwater than saltwater. And that's because nitrite has this -2 charge on it, which so does chloride. And so the gills actually don't know if it's chloride or uptaking nitrite. And so the blood is a lot more healthier if you have chloride there, where they can take up more oxygen rather than inhibiting it with the nitrite. So for this case, adding salt water will actually be more of a protection for the fish, because it'll block that uptake of nitrite. And so really anything over 2 milligrams per liter of nitrite is unsafe and can lead to really bad health problems. So what does the nitrate, and I'm sorry, I'm not going to talk too much about that right now due to time, but nitrate is more of a toxic form in saltwater, but it's really one of the safest forms for nitrite in freshwater systems. And it's commonly talked about. And so when we talk about clean water, these are given concentrations, though, like I said, it's just a snapshot of a continuous cycle, because the nitrogen's always there in some form. It's just what form do we want it in, and or do we want it uptaken by plants or by the fish? Where is it going, and following that flow. The other one that we keep talking about now with nitrogen is phosphorus. And most phosphorus is actually in particulate form, and it's mostly bound into sediment. It's very difficult to keep phosphorus in the water for that's readily available uptake, and that is called soluble reactive phosphorus. So that's available phosphorus. Phosphorus and nitrogen actually have a linear relationship. This is a wonderful Redfield ratio, and so typical ratio is usually 5-to-1 or 20-to-1 in terms of what a living creature needs to continue to live. And so marine plankton needs some silica, and so that ratio kind of depicts what algae are present. It's shown harmful algal blooms depending on a more narrow ratio, for instance. And so phosphorus, as we increase our phosphorus, which we have found has been the limiting factor in a lot of growth, now we're increasing the odds that the plankton are changing and creating off flavor, and so that those kinds of effects. But we want to be able to supply phosphorus for the system and that it needs, and it really will, for that reason, will cause a greater response in plant growth than it will increase in nitrogen concentration. And so nitrogen is in that cycle, but phosphorus is really bound in sediment or plant growth. And there are studies out there that show when you add your fertilizer, nitrogen or phosphorus is typically taken up into the sediment within 24 hours. And so solids are the next kind of big parameter, and it really can result in high rates of turbidity and sedimentation. Pros and cons to them depending on your system that you have, but it can really affect shallow areas more because it destroys the benthic organisms if you rely on those and fish eggs, and you can use it. So sometimes you want light penetration or you want to shade from light, right? So you can control the light based on turbidity, but this also can limit production of phytoplankton or other aquatic plants. And it detracts turbidity. Really, it doesn't, if we see turbid like this system here, it doesn't look clean. And so humans are more looking for recreational purposes, or that, we want clear water more for the aesthetic unit. But suspended particles also create a crust. So there's quite a few things in terms of solids and maintaining solids. Particulate matter can be what's going into the water, such as feed. You don't really want the feed to sit there for longer than 10 minutes. That means you're overfeeding. You want to remove that feed because it's also creating an oxygen demand, or that sludge builds up over time, because if your aerators are constantly running, they're stirring up sludge, they're creating erosion, and all that turbidity is being expelled into the environment, causing those impacts that maybe we don't necessarily like. And so overall, one simple management decision can have a downstream effect. And so I would just kind of re-emphasize, which is probably going to be my go-to lingo now, is that clean water makes for a happy fish, and happy fish or healthy fish or happy fish. I'm going to get that right one of these days. But with that, I'm happy to point you to more questions. It's really hard to take a deep dive. This could be a whole entire class that, we sit down into each parameter, but this provided a brief overview. If you want a specific chapter to kind of read more about, these have been some of the best resources that I have used, although interpretation and doing the math is sometimes very complicated, as I'm sure Boyd students know. But with that, I really want to thank you guys, and I'm happy to answer any questions. Thank you.